The Michelson-Stahl experiment is an experiment by Matthew Michelson and Franklin Stahl in 1958. It supported Watson and Crick hypothesis that DNA replication was semi-conservative. In semi-conservative replication, when the double-stranded DNA helix is replicated, each copy contains one original strand and one newly synthesized strand. Nitrogen is a major constituent of DNA. 15N and 14N are two isotopes of nitrogen. The DNA of the cells grown in 15N medium had a higher density than cells grown in normal 14N medium, which can be distinguished based on their densities by centrifugation in cesium chloride. Here you can see DNA from cells grown exclusively in 15N produced a lower band than DNA from cells grown exclusively in 14N. Michelson and Stahl conducted their famous experiments on DNA replication using E. coli bacteria as a model system. E. coli was grown for several generations in a medium containing heavier 15N isotope. After many generations growing in the 15N medium, the nitrogenous bases of the bacteria's DNA were all labeled with heavy 15N. E. coli cells with only 15N in their DNA were transferred to a 14N medium. E. coli doubles every 20 minutes. DNA isolated from cells at the start of the experiment produced a single band after centrifugation. This result made sense because the DNA should have contained only heavy 15N at that time. DNA isolated after one round of DNA replication also produced a single band when centrifuged. However, this band was intermediate in density between the heavy 15N DNA and the light 14N DNA. When second generation DNA was centrifuged, it produced two bands. One was in the same position as the intermediate band from the first generation, while the second was higher, appeared to be labeled only with N14. This result told Michelson and Stahl that the DNA was being replicated semi-conservatively. The pattern of two distinct bands, one at the position of a hybrid molecule and one at the position of a light molecule, is just what we would expect for semi-conservative replication. In the semi-conservative model, each hybrid DNA molecule from the second generation would be expected to give rise to a hybrid molecule and a light molecule in the third generation, while each light DNA molecule would only yield more light molecules. Thus, over the third and fourth generation, we would expect the hybrid band to become progressively fainter and the light band to become progressively stronger.